So, good afternoon, guys. In the today's lab experiment, our aims to measure and analyze neural responses in the auditory pathway using brainstem evoke response audiometry, which is also known as BERA. So, let's get started. So, here is the introduction of the BERA. BERA stands for brainstem evoke response audiometry. It is a non-invasive technology which is being used for measuring the auditory signals from ear to the brain. It is used to evaluate the auditory nerve pathways up to the brainstem. This evaluation allows us to understand how the auditory signal reaches our brain without any with the in the auditory pathway. It provides information about hearing and neurological function. Here is the BERA test device which is being used for this experiment. Next is the history of the BERA. BERA was developed in 1970s as a tool for assessing auditory function. This tool is used for assessing the auditory function in the 1970s. It is usually performed on patients who could not participate in conventional hearing tests. Now it becomes the standard diagnostic tool in auditory and neurology. Next is the working principle. BERA basic working principle. It works on a it is an electrophysiological test that measures the electrical activity generated by the auditory nerve and brainstem in response to sound stimuli. Here, the electrodes are placed on the scalp to record electrical activity in response to the auditory clicks and tones. These are the input signals in the form of clicks or tones. Next is how the BERA works. Here is a stimulus delivery system. In that auditory stimuli, there are clicks or tone bursts that delivered through headphones to the patient's ear. Here in the diagram, as you can see, there are four electrons being placed, one on the forehead, which is ground, second, which is on the scalp, is the reference electrode, and two electrodes near the ear lobe is active electrodes. So here, the frequency and intensity, the stimuli can vary in frequency and intensity to access different aspects of hearing. Now, next is the electrode placements. The electrode mostly placed on scalp and earlobes. Electrodes are placed on the scalps and earlobes to capture the electrical response. Second is a ground electrode. The ground electrode is also placed to reduce electrical noises. Now the electrode placement, reference electrode which is being placed on the head position, which is CZ position according to the 1020 electrode system of EEG. Second is ground electrode placement, which is on the forehead. And third is the active electrode, which is placed in the mastoids behind the ear. The waveform analysis. The waveform, the auditory stimuli generated electrical responses in the auditory nerves and brainstem, which are recorded as waveform. BERA generates a series of waveform 1 to 6 or 7 between the first 10 millisecond of after the stimulus been applied. Each waveform correspond to specific anatomical points along with the auditory pathways. As you can see in the diagram, there are 6 7 waveform being developed from that test. Next, the waveform analysis. There are 1 to 7 waveform that are generated from different different brain parts, which are cochlear nerves for responsible for wave 1 and 2, cochlear nucleus, brainstem, which is responsible for wave 3, superior olivary complex, which is responsible for wave 4, lateral laminices for wave 5, and inferior colliculus midbrain for wave 6 and wave 7. Uses, it is mostly being used for diagnosing hearing losses in patients of all ages, including newborns, accessing auditory nerves and brainstem function, detecting neurological disorders affecting the auditory pathways. That's my references for this presentation. Thank you.